getting data at least. Yeah,
Wow,
that's
that sounds
helpful.
okay. Wow, that sounds very good. Very super.
Yeah.
Hi, Bradley.
Hi, how are you?
I am good. It's been really long. I haven't seen and joined.
Yeah. Yeah. Well, yeah. Hopefully, you can give us an update this week and see where we are. And yeah, Susan, hello. So yeah. Well, yeah. Let's start. So yeah, why don't you start, Mehul, and then we can go to Susan, and then hello, Morgan.
Okay, I'll share my screen.
All right. Yeah, it's good.
So Bradley, so last time in the discussion, Bradley and I were discussing data, which was C. elegans data. Essentially, it was this. Let me open that data also, so it's good to understand. That in the name is C raw data. It should be found in data C raw. Yeah, so I'm opening along with it. Okay. So this was the data last time Bradley and I were discussing, and Bradley actually gave me a lot of insights in detail and helped me understand. So we have here cell column, time column, XYZ is the coordinate of that particular cell, and size also evolves along with time, as we can see. And yeah, and then Bradley actually directed me. He told me that I can find lineage data also. So I searched on OpenWorm website, and I was able to find it, and I have used it for finding a daughter cells and parent cells and all. So mother cells, yeah.
Yeah.
So in the discussion today, I plan to cover the data understanding, what I understood from data, and then I have done some data preparation, basically trying to prepare it for the graphs. Then I have made a graph data based on the spatial data. So I plan to create graphs for spatial as well as cell lineage, but right now focusing on uh, uh, the spatial graph. Then I also created a spatial hypergraph as well. I have a couple of ideas to create, and I have shown both the ways. If we finalize with one, we can go with the that one. So I'm proposing the two ways. Then I have other ways of creating hyper edges based on the lineage data. So I have a couple of ideas, and then in the end, introducing the dynamic graphs. So that is actually the last part, but still keeping those things already in mind, we will design everything. So I'll start the discussion.
All right.
Okay. So in data understanding, so this is how data looks like. We have cell, time, XYZ columns, and size. And I was just reviewing the data. So it is time is an integer, float, and XYZ is coordinate, so it's float, and size is also in float. So I just saw the stats of the data. So count is like these many data sets, these many points. These are rows. So for every time, for every cell, we have a recorded data. That is why this is so large. I think it's 250 to 100. So that's quite large. And then we have XYZ column. So we have mean of it, something like this. I will tell how I will use this later on. And then for size also correspondingly. Then we had one interesting time column. So I wanted to see what time column looks like. So we have time from 1 to 180 190 actually, and this is a contiguous data. I think some are missing. Actually, nothing is missing from 1 to 190. I saw something missing in between, but yeah, right now I'm not able to see it. I hope it's continuous, and probably you can confirm from 1 to 190 time point. But there was some negative points also. So minus 16, minus 15, minus 14, and minus 30. So I saw which particular cells belongs to these time points. So those are P1, or actually P1 only, and their size changes little bit across the time. So I was not sure how what is happening here. But I think this can be noisy data probably. But because just because I was not able to understand, but probably it might be conveying something. So coming next to size of cells. So just to observe what is the min and max value of size. So the minimum value of a cell is 24 units. I'm not sure. I think it's micro, some micro nano size, of course. And the max size is 103 units. And then we have frequency of count. So the total number of unique cells are 786 in this particular data. And I just did the value counts of it. So trying to observe how many times a particular cell is observed. So this kappa, CAAPA, is observed the most number of times. So I printed the top 10 which are observed. So this observations is basically number of times it comes and number of the frequency of that particular cell. So multiplied by those, we get this 806. So to understand this better, I plotted this corresponding to time also. So when we see, let's say this is a new cell, EARA. This cell occurs at the 10 time, it occurs 14 times. That means there are 14 instances of this particular cell at the time 102. And also at 101, it has 14 instances in the organism. So this is how it looks like. So I think this gives me a good understanding of data. This gives us good understanding of data. And then, yeah. So then I wanted to map it along with the lineage data as well. So in this data, we do not have the information who is the daughter cell of which particular cell, right? So for that, Bradley pointed me in the last meeting towards this particular data set. I downloaded it. So it looks like this. Yeah, I hope my screen is changed now.
Yeah.
So we have cell name as this, A, B, all these things. Then we have location. So location is nucleus for everyone. I checked it, so not required. Then we have object, subclass of. This is also a not very useful column. And then relation. So relation tells us daughter of. So basically this is the cell, and P0 is the mother of AB, and AB is the mother of ABA. So that is what it conveys. So using these two particular columns, we can see this columns here. So this is the cell, and these are the mother cells of it. Yeah, and then I wanted to see how many data points exist. Actually, I will go more deep into it. I didn't do very much exploration of this particular data. So there are 1,800 rows. So I don't know why it is more than 786. Not 786. The previous data we had how many? We had 786. Yeah, 786. But here we have 1,864, which I am not sure about, but I think it doesn't matter at this point for us, so I didn't explore that much. Right?
Yeah.
So these were so this is about data understanding. Now coming to data preparation. So now what we want is we want to make two types of graphs basically, or hypergraphs. One is spatial and one is the lineage. So for the spatial, we have to first little bit normalize pre-process data. Do something with the data. So Bradley suggested in the last meeting that we should normalize the data at the 0th point. So that is what I did. I calculated the mean of all the axes, which is x, y, z, and then subtracted from all the points. So these three x, y, z columns are little changed now. Just the normalization is happening. Not normalization exactly. I just subtracted the mean, but but I can also do normalization if required. Actually, I thought this is okay, but if required, I can do normalization. We just have to divide by the max and min value, max minus min. But still, I haven't done it, so I'll just comment it. Then, what is this? So then basically I wanted to include this particular the lineage information in the same data as well. So I did some code, and this is what we have in the end. So we have the final data which we are going to use. So we have here is cell information. Oh, it is not yet ready. So I've added lineage. After adding lineage, this is how our data looks like. So this is the cell information. This is the mother of each cell, and then we have this XYZ. And then I thought of making the min size and max size of every particular cell, because we had a lot of timestamps, right? So I thought min size and max size would be useful. Probably we can take more features, for example, average size also. So we can add them as required. So when I printed the size of this data, we have 780 columns. So 780 rows. I'm sorry. So one row for every unique cell. So this was the data preparation. But I noticed one thing. So initially we saw that we had 786 rows, right? But here we are getting 780 rows after combining with lineage data. This is because I saw what are the missing columns in the lineage data. So in these these cells were missing the lineage. So probably because P1, P1, P2, P3, P4, Z2, Z3 doesn't have any parents. So that is why it was missing. So what I did was I included these 6 data points also as rows in this particular data, but I renamed their mother as Nan. So just to remember that these particular cells don't have any mother cells. So these particular points. So what I did is I concatenated it with the previous cell. So we have now the new stats DF, the data frame we are going to finally work on to create the graph. So any particular point still here, or should I complete everything and then we can discuss?
Oh yeah, let's go through the whole thing first.
Okay. Then I was on graph creation part. So we need 2 types of graphs. So the first is we need a graph created from the cell positions, and the second is graph created from cell lineage. Referring to the ESOP 2022 by Jihang Lee's project, so what he has created is he has created a graph using the cell lineage. And cell lineage, since it gives information from mother to daughter, he made a directional graph because there is directional information involved with. So, but we don't plan to yet use directional graph because I'm not sure how hypergraph can be directional. But we can think about it at a later point. So, but sticking to this particular point, sticking to the first graph, the cell positions, we'll start with that. So I have used torch geometric to create the data, to create the graph. So I have used KNN graph. So KNN graph is basically based on the coordinates of x and y. So this is basically graph creation. What I have done is I created a fully connected graph. That means every cell connects to every other cell. Basically, they have an edge, but the edge value will be different. The edge value will depend on what is the distance between those cells in the coordinate system. Yeah, and actually we can threshold also, basically based on how many distance we want. So I thought this is the most general graph we can create at this point. Later on, if we want particular things, then we can change this fully connected graph itself, right?
Yeah.
So this is how our complete data looks like. We have 786,3. So 786 is the number of cells or the number of nodes, and 3 is the number of features. So in features, I have included the size, which is min size, max size, and there was one more thing. We have also mother information here as the node information, and we also have the name of the cell. So these 3 information is incorporated in this particular X. And then we have edge index. So these edge index specify the shape, has a shape of 2, 617,000. So these are basically number of edges. So in this sense, it's a fully connected graph. That is why the edges are so many. And here 2 represents basically that it's a pairwise. It's a pairwise graph, pairwise edge. That means 2 particular nodes belong to it. And then we have edge attributes also. Edge attribute is basically the weights between those particular nodes or the cells, right? And these are just the node size, node names, and mother names, just incorporating all the information. Then I tried to visualize this graph. Which was a bad idea. I tried to visualize a fully connected graph, and it took, I think, 25 minutes to run. Then I stopped in the meeting. It was way too tiring and not happening. So instead of creating the fully connected graph, I just visualized the 2D, only the X and Y. So this graph doesn't tell anything, but just for the visualization, I try to plot the cell positions here. It doesn't convey anything, and that is what I wanted to show, that it's not conveying anything. Right. To visualize a graph, we have to threshold and then we will plot something. Yeah. So this is the graph creation part, but our main task is spatial hypergraph. This was just a simple exercise, probably to get ideas on all. So now coming to spatial hypergraph, we have two ways to create it. So first way is we can use a previous graph to create the hypergraph. So I have named it as distance threshold based approach. So thresholding is basically what does it mean? Wait, okay, so here I am giving 50. Yeah, so here we are specifying threshold. So threshold I will give default as 50. So what we want is we want to create many hyper edges, and each hyper edge we are specifying how many cells are going to be there. So that is a threshold value. So let's say we will be creating number of hyper edges will be same as the number of cells, which is 786. 786. So if threshold is 50, then we will be creating 786 hyper edges, and each hyper edge we have like 50 cells, which is a lot. So I will show this is not a very good idea, and we will have to reduce it. So what I have done is based on the distance proximity, using somewhat like KNN. KNN is K nearest neighbors. We have created a graph with threshold value of which is the KNN hyper edge. So this is how it looks like. Pretty hard to understand, but what it says is in the lowest most circle, so we have different colors for different hyper edges. So in the lowest most circle, there are like some few nodes, and as we go, let's say in this pink one, we have like a lot of nodes. So this is a basically bigger hyper edge. And these are the hyper edge numbers, randomly given numbers. So this really honestly is not telling anything. So what I did was I reduced this threshold value to a lesser value, which is I think 5. 5. And then we plotted this graph. So I think this looks little more sane and little bit probable to understand, but still I'm not satisfied with this, but we'll go to next. But just understanding this particular one, we see that we have a lot of hyper edges, and each hyper edge has like 5 number of nodes here. So this looks very dirty in the beginning, but I have made a bigger one and downloaded, so I can show it at a later point also to understand in more detail. So this is basically the first method by thresholding based on the distance, which we are using utilizing the previous graph itself. And now coming to second method, that is actually little better. I wrote somewhere the benefits on paper, but I can't find that paper now. Here we have this method is basically using the clustering approach, and I have used DBScan clustering because DBScan it has some benefits. I forgot. I will mention them later in the meeting. But I wrote like 3-4 useful why I went ahead with DBScan and why clustering based hypergraph is little better. So here, what we are giving in DBScan is we have given 2 hyperparameters. One is EPS and one is min samples. So EPS means the distance, the max distance basically. So for example, a hyper edge, two nodes have a distance of let's say 4.5. Then they are going to be in that hyper edge. If that nodes is farther, let's say 5.5 distance between 2 nodes, then these two nodes are not going to be in a hyper edge. And to be a hyper edge, there has to be minimum like 2 samples. There cannot be a hyper edge with 1 sample. I mean, that we can change also. I have changed it later on and I will show later. And so I wrote a function to create it. This is the function. So the hyperparameters I have given is EPS, min samples, and include noise. I will try to just tell what it is after show. So here we see that we have a much cleaner graph. So here in the previous threshold based method, we have to make sure that all the hyper edges had like 40 nodes, right? Or maybe let's say 5 nodes. But here it is not the case. Here it depends on the density. So DBScan basically, I think it means density based something. I don't remember what the full form is. DBScan. That's true. So it's a clustering method. So it basically creates lesser number of hyper edges. So since and here this include noise basically I have used as true. So include noise basically means there were a lot of nodes or cells which were not coming in any particular hyper edge. They were not able to belong to any particular hyper edge. So if when I set it to true, what they can do is they can create their own unique hyper edge. So for example, only that particular node or that particular cell will be in that hyper edge. I don't think that is a good idea, but just for trying, I did that. I also tried with setting it as false. That means those cells are not being included in the visualization itself. So that is this diagram. So this we probably from visually we can see it has lesser number of nodes because we are removing the noise cells from here, which are like very far from every node. But here we are losing some data, so it has some pros and cons. Yeah, so this is actually we can see it in much more detail, like if we focus on particular areas, which we will do. But I wanted to highlight the benefit of this DBScan based hypergraph creation or clustering based hypergraph creation is. So by the way, just a note, here we only have I think 116 hyper edges compared to the previous method where we had 786 hyper edges and it looked very cluttered, right? So that is the one benefit of this clustering based spatial hypergraph creation, that we have less number of hyper edges. That means less overlaps and less noise. I mean, if we have way too many hyper edges, then information propagation will happen a lot faster. Information propagation, or we can say diffusion, and sometimes that much diffusion is not required because it can do over smoothing also. So it can contribute to over smoothing also. So I think we should do a balance between these two, or probably I think this is better if not the balance stuff. But visually, if we try to see, we can make sense of it if we have like a bigger figure, which we will see in the end. Yeah. So this is about. Okay, this is something I tried. Okay, yeah, this is the false one. Yeah, so this is all I tried about the spatial hypergraph. Probably if we are good with it, I can create a proper API of it for like pushing the code and all. This was just the research aspect, and probably if like Bradley suggests, or if like you guys, if everyone suggests, we can probably dive deeper into particular things and explore more ideas. I have some ideas, but these two are like some good ideas which I felt we should try. And if like in the end we can go ahead with creating the API, whichever method we found the best, or probably we can create APIs for 2-3 ideas which are good. Yeah. Now coming to lineage hypergraph. So this is not was I planned to do till now. Right now the plan was to do only spatial, but yeah, I wanted to discuss because it's coming up in this July month. So this yeah. So how do we create lineage data? So one approach we can do is we can create hyperedges based on common parent. So for example, if let's say a particular there are 5 cells and 5 cells have a common parent, so those 5 cells can be part of a single hyperedge. So just an instance. This was my criteria for creating the hyperedge in the lineage data. So I have created using this only, and this is what we can observe. So I have a. So here we can see that here we have the name for the hyperedge also, and we have the name for the nodes also. So black ones are the nodes, and the different colors are the hyperedges. I can draw a much better figure. I am using some library called HyperNetX. I found it at some point during. So this is actually very good for visualization, and I can change it to better colors and all. This is a little more flexible. So just to observe this data in more detail, but it didn't take a lot of time to visualize. I think it took 20 seconds to make this large image. So we can make even larger also.
Yeah, it looks good.
Okay. Yeah, thanks, Bradley.
Yeah.
And coming to last point, just last point, then we can discuss. So then it comes to a dynamic hyperage, which is our last plan, which we want to do. But still, I think we should plan things from now on, so we have a good understanding and things go very smooth. So as we observed before, like we have time points from 1 to 180, 190. We have like contiguous time steps, right? And then we have some negative values. So I was thinking whether we should create spatial graph and lineage graph for these particular data points, basically for all data points. But let's say for the seventh time, there are only like, let's say, 5-6 nodes existing. So we create nodes only for those. We create graph for only those particular cells that exist at the seventh point, and then we are going to have some graph something like this. So this is at different time points. I mean, this is just for the idea. This is some other data set and some other thing. But probably we will have some evolving graph at all the times from 1 to 190. Or if we think that 180 for 90 is a lot, then we can threshold. We can do for every 10th time. But yeah, whatever sounds good and whatever gives more clarity on understanding the data.
Yeah. Yeah. So that looks good. Thank you for the update. It was very good and very complete. And you produced a lot of visualizations, which I love because it's we can see what a hypergraph looks like, and from the data. So I mean, one of the things about the plots is that we don't, we can plot like every cell, but we are often interested in the sort of the cells that are alive at a certain time. So, if we think about the hypergraph as being present, say, at 180 minutes, like what cells are there at 180 minutes? And then maybe what cells are like the mother cells of those and the daughter cells of those. So I think that might help with like creating visualizations where you just would build an all-to-all graph or a hypergraph, and then maybe have one where you take a couple time slices where you go through like at certain times and say, okay, at this time, there are certain cells that are alive, certain cells that are the mother cells, certain cells that are the daughter cells, and that's it. So you have like 3 sets of cells, and then see how those are connected. Because what we're interested in ultimately is, we're interested in all the relationships, but to see like at a certain point in time, what the mother cells are, what the current cells are, and what the daughter cells are. So it would be like the current cells are basically the spatial distribution of that level of the lineage tree, or the cells that are alive at that time. And then the mother cells, of course, are interesting because they're the ancestors, and so they're passing things on to the daughter cells. And then the descendants are interesting because those are what are going to be born, and then you can say things like, okay, how do those move in space relative to the current cells, which would be the mother cells for those cells. So you would have this nice 3-layered graph, in terms of the lineage tree, and then that would be the spatial distribution of those. And you could say a lot of interesting things. You could say, okay, a mother cell divides into 2 daughter cells. Those cells migrate out. Then the daughter cells of those migrate out. And so you have like this 3 layers of the lineage tree that are represented there. And you can say, okay, now what are the hyper nodes for those cells? And and that would be like, because I think you have, especially the last 2 hypergraph visualizations were clean. I mean, I think the ones that are clean, where you can see the hyper edges or the hyper nodes clearly. The hyper edges are a little harder to interpret because I guess the question here is like, if we visualize the hyper edges, it's like technically 2 hypernodes are connected, right? And then the hyper edges can be heterogeneous. So the hyper edges can be connected to, say, like multiple hypernodes, theoretically. Because if we're using a clustering algorithm, some of the cells are going to be, connected to other cells in another hypernode. but maybe another hypernode as well. So you can have this sort of heterogeneity. But this is nice. Just being able to define the hypernodes with their contents, and then having these edges. And I don't know how to I don't know how to visualize these really clearly. And this is just something we're going to have to work out. But those visualizations, I think, are going to be important because, ultimately, a lot of these network science things, there are two things that are really kind of are the outcome of a lot of these studies. One is like the visualization, and a lot of like network science papers will just show like the hairball, and they might try to extract some communities from those, and it's hard to really visualize everything and get it's like you see the visualization, that's one thing. Understanding the sort of the causal relationships, that's another thing, and I don't think people have mastered that at all. The second thing, of course, is the content within so you're basically looking at these all-to-all representations, and it's like if I just take a connectivity matrix and dump it out into a visualization, what do I get? So we want to take like maybe the most informative slices of the data and then plot them out, and that can help go a long way towards sort of getting us to a point where we can say, okay, we're going to reduce the data down somewhat because we can't just put everything to everything and go for it. But yeah, I think we should think about like these informative slices of time where you have like a subset of cells, daughter cells, mother cells, and then the current cohort of cells. And so yeah, and like the paper that we did on spatial networks, just the regular networks, you plot these out, and there are a lot of things that are interesting about all cells, kind of at all levels of the lineage tree. But really, you just want to get the ones that are alive at a certain point in time, and look at those. So yeah, there are a lot of interesting things you can do here. This is really good work, and I like some of the Python toolboxes you're using, and I think the dbScan method is good. And there are always going to be drawbacks. I think what would be good for the sort of for GSOC, and then when we'll be writing this up as a paper too, would be to sort of have some documentation so that you can like say these are the methods that were used, these are the toolboxes that were used, these are the pros and cons of each toolbox and method. So like, why did you decide on dbScan? You said that there are pros and cons. Why did we not? We tried this other method, and that has some pros and cons. So if someone wants to say use this method, they can do so, but it depends. Like if we're looking at C. elegans, maybe this is best for C. elegans. If we wanted to apply this to Drosophila, maybe there was a better way to do it, and there are reasons for that. And then of course, the toolboxes that we're using here, are, I guess, they're open source, so we need to attribute them and cite them and everything. So I want to have like some documentation with this as well, and definitely it'll help us later.
Sure. Okay. So the next things I will try to do is I'll just jot down the points. So,
Okay.
the documentation for all the steps which we want, documentations of decisions, about libraries and methods like dbScan.
Yeah.
Right. And the immediate next steps, what else can I do?
Next steps, would be to kind of, like I said, go through the data and pick out subsets of the data that are interesting. So maybe like all cells that are alive at time point 200, take the mother and daughter cells of those cells, and then use that as like a subset. And you can like subsample your data set in different ways, and just test out different subsets. And maybe ideally we would like to have a graph or like a report that says, this is the hypergraph at 200 minutes. This is the hypergraph at 300 minutes. This is the hypergraph at 250 minutes. And it's going to look different. It's going to evolve. So the idea would be to have like a hypergraph that evolves, and you can see over time as the embryo is changing what that hypergraph how the hypergraph changes. And it's up to you to see what that looks like. Yeah.
Okay, yeah, thanks. Yeah, that actually makes that's a very good idea. I'm definitely gonna try this. Bradley, about the spatial graph. So should we go ahead with creating? This. So we have tried these two methods, right? Distance threshold based approach and this clustering based. So should we start creating it into API like Jihang has used and like PyTorch or PyTorch geometric exists? Basically, another person can directly utilize it. Should we do it at this point of time, or should we push it for later and right now do only the exploration part and documentation part?
Yeah, I think we'd save the API for later. And that's like, this is good, and just keeping on this and trying to get this down to like where it makes sense and we can get some good visualizations. And then hopefully later we can do some APIs. I don't know what Jiahong had in terms of an API, but maybe we can, kind of use the same one or use a similar one so we can integrate that. So I don't want to like have, two different APIs for the same project. I don't want to use, I want it to all kind of be one thing, but I think that's something for like later.
Right, makes sense. So he has the API basically for bidirectional graphs, which was incorporating lineage information.
Yeah. Well, yeah, I mean,
okay.
Yeah. So, I mean, the idea would be someone could download maybe like, I don't know, like, basically they don't have to like be confused as to what to use for their problem. So if we need to create it like maybe like a compatible API, so like we could take like, we could tell people, okay, if you want to build a bidirectional graph, this is a tool. If you want to build this hypergraph, this is the tool. Just so that it's easy for people when they fork the repository to sort of find what they need.
Yeah, right. Yeah. Okay. And Bradley, last point. Apologies for missing out a lot of meetings. So, but from now on, like every week, I'm going to show some updates because for a very important reason, because I think this is more of like research, and I need a lot of your feedback every week. So that would help project run very smoother, and also decide at every particular steps why are we taking those decisions. So yeah, I wanted to mention that.
Well, yeah, that's good. Thank you. That's a good update, and I look forward to kind of getting this off the ground and getting some really interesting stuff out of it.
Right.
Yeah.
One last question. Okay. It's a technical one actually. So I'm going. Sorry, you have to repeat this particular point. I think we have discussed this before, but I can't remember it. So what is the difference between hypernode and hyperedges? Like just in technical terms. So as I understand, hyperedge is a collection of nodes in a way. But you have mentioned hypernodes a lot of times in the paper, right? So I didn't fully understand what is the difference between these two? Are they like same things?
Well, hypernode is a single node that has a bunch of nodes in it. So like in the clustering method, you might have like 10 nodes that cluster as a hypernode, right? So you might have like the content inside the hypernode are individual cells with identities. And then those things are kind of clustered together. And you can refer to those collectively, or you can refer to those as the individual nodes. And then the hyperedges are the things that leave that hypernode and go to different hypernodes with their own cells in them. And so this is why, you have hyperedges, which are like a bundle of nodes going from one hyperedge to another. But that means that you have hyperedges that go to different nodes. So like that's the idea, that it's almost like you have like a super graph, and then inside that super graph you have parts of regular graph, and you're kind of trying to bundle those things together in the way that so they share like a connection. If a bunch of nodes are connected to a bunch of other nodes in groups, then those are all bundled as a hyper edge. and then the cells that are clustered together, bundled together as a hypernode. That's basically the way I think of it. Yeah. And then, it's kind of tricky because it's hard to wrap your head around because you have this regular network and then you have this other network on top of it. But yeah, I don't know if the tools are making, I don't know if that's the same way they treat those in the tools that you're using. But I mean, yeah.
Okay, yeah, that makes sense. That makes sense
Yeah.
a lot. But just for the tool's sake, actually in the tools I was using for visualization, just to give them credits and highlight them. So I'm using this HyperNetX. They have a paper also in 2023, I think in some conference. So this is a very nice tool for a lot of objectives, for basically visualization, of course, and then it provides a nice way to create hypergraphs and then do basic arithmetic operations also. So and one more thing was there. So it was related to Pakhi actually. So that's why I wanted to highlight. So it can help us understand the topology of hypergraphs as well. So it has some metrics. If I'm not wrong, I was seeing some in one of these collab notebooks. So it's a pretty good package to explore for later on understanding those hypergraphs. As we were thinking, we have a big hypergraph, but we want to understand it through some metrics. So this library can help us in some ways at least.
Okay. Yeah, that's good. This looks, like a nice toolbox. And they have tutorials and everything, so that's good. And I like, what are these, these drawings up here? So these are like paths. Yeah. So these are like clusters of nodes that they show up here. Yeah. So this is like a nice example because it's basically taking the nodes and grouping them in different ways. It's like you have Slytherin, Hufflepuff, Gryffindor, and like it can be nested or they can be separate, and then those are connected. So in Slytherin, there are 3 nodes. Those are connected to Ravenclaw. There's an overlap in this case, and so they can overlap certainly. But then you have other nodes that connect to other nodes in other hypernodes, and so those are the hyper edges. And so yeah, this is a different way of visualizing it because they consider overlap between hypernodes. But you can think of hypernodes as categories that you fit your nodes into, and then the hypernodes are connections between those categories.
Right. But this library hasn't highlighted hypernodes anywhere, as far as I remember.
Oh well, they
I don't
know what the terminology is. I mean, people are like, this is kind of an area where people have different ways of thinking about it. So, and it's hard to really kind of get around. Like, yeah, but basically, they're trying to classify nodes in different ways. So, yeah.
Right. Yeah, so thank you very much. That is from my side.
Well, yeah, thank you. Yeah, it was very good update. All right. So, Pakhi, do you have an update? In the meantime, why don't I highlight her the pull request she made on the repository? So, this is her pull request in the DevoGraph repository. This is the K-mapper implementation. So this is, and I think she said she was going to make another pull request soon, but this is with the K-mapper implementation and some other details. So this is the fork of the K-mapper toolbox and some of the things that she's been working on with respect to that. And then she's merged it into DevoGraph. So congratulations to Pakhi for that contribution. And it looks like she's back.
Yeah, am I audible?
There we go. All right. Yeah. Thank you.
Yeah. So I've been working on like this NDP function. This is almost complete. I just need to integrate like the functions here with the other like files. So in their repository, they have like a very organized framework. So first it will call like the train.py file. It will then call this one file. In this file, it calls like the MLP function and the adding new nodes and the predicting new nodes. And it also calls like the evolutionary algorithm. I'm also working on this one. I need to like make some changes to this. So like this integration part is taking some time. I also have like my exams going on right now, so I wasn't able to give much time to the project. But like this file is almost complete, and I just need to like integrate and then train it. It works, but it's showing that the graph is disconnected. That's probably because in their architecture they are generating everything randomly. Like the initial graph is random, and then the new nodes that get like added to other nodes are also random, and then they like check if it's follows like the small world network topology. But like here in our case, we have like the data set available and all that, so I need to make those changes. And if like in the last meeting you suggested like the small world network is similar to like the growth of C elegans is what I understood. So I'm trying to like use the same functionality here as well, just modifying it to our usage. But if that doesn't work out, I will like go with my initial plan of like making the whole architecture like from scratch. So I'll give more time to this because I think I can like try to implement this only. I would like to show how it would look like. So this is the small world network that they have generated. So once I'm able to train it, then this is how it would look like. So this is what I'm trying to do right now. This should take like a few more days, 2 to 3 days, and yeah, I can update you after that.
Okay. So yeah, it's just generating like nodes and then edges with as the nodes are added, and it's doing in that small world fashion, correct?
Yeah.
Okay. Yeah. Yeah, I mean, yeah, there are going to be some challenges because we can't say that C elegans is a random network. I mean, I mentioned I think last time about how the connectome is small world, and we might assume that the sort of the larger graph of cells is also has some structure that is maybe small world, maybe not, but small world is a good heuristic, and then we can work from there. But I don't know like how it's handling the incorporation if it's doing it at random. That's maybe not going to fit the data we have for C elegans because it's not really the cells are you have the mother cells and then you have the daughter cells, and the daughter cells migrate in a non-random fashion. They migrate either they're left-right pairs or they move anterior posterior from the mother, but they're not just generated at random. There's some contingency there, so that's the thing. And I don't know what you'll need to do to make that happen, like I don't
Yeah. I have some idea in mind. Like in the paper also they've used like 2 3 MLPs like for placing randomly and like generating randomly and all. So I don't think we require all that because we already have the data set. We would need only one, and it will like predict, okay, like maybe here the new node, the child nodes get added, and then I can like compare it with the data we have and then back propagate the losses in order to like train the model. So that's what I'm planning. So I'll let you know once I'm able to train it.
Okay, well that sounds great. Thank you for the update.
Thank you.
And so yeah, in Mehul, please issue a pull request for the work that you've been doing when you can. Whatever you think is a good sized package for the pull request. We want to keep everything up to date and backed up and everything. So that's good. Thank you.
Okay.
Yeah. All right. So
Bradley, so for the pull request, yeah, I'm sorry to
okay,
interrupt.
go ahead. No. Go ahead.
Yeah, so in the pull request, should I upload this Jupyter notebook, is it?
Yeah, please push the Jupyter notebook and any files that you have. Like you can push image files as well and just put them kind of put together the repository so that it has, maybe a README because you'll be pushing to this again. You want to have like your space and then you want to have your, you'll issue a pull request and then you'll update with new files. So please, the notebook, images, maybe a small README now for now, and then we'll add documentation as we go. So
yeah, makes sense. Yeah, I'll do it.
Okay, so I don't know, Susan or Morgan have updates. Yeah, we have some things in the chat, and it's largely just kind of the Pakhi said, forgot to mention, thanks Mehul for sharing the visualization library. I'll try using it too. Your visualizations look great, so that's good. Please share that with Pakhi about, where to, I don't know if that was a thing you were highlighting or if it's a different package, but yeah, it's good. Okay, so yeah, Susan, I guess you want to do an update.
Oh, well, I think I mentioned before, I've been accepted into CompuCell.
Okay.
The CompuCell 2D and 3D course, and so I've been preparing for that. I have to program in Python, and of course I haven't. But I'm assuming it's C-like. People say it's C-like, and it's much like MATLAB or C, and I have programmed in that. So I'm just kind of preparing for that, and I discovered that my lovely console program was exaggerating the movement of the parts by 10 to the minus 5, so that's why I was getting these odd numbers.
Yeah.
They were off by 10 to the minus 5 for some reason, yeah. So I found out where to fix that, and now I'm getting proper numbers, at least. There's still a few problems, but at least I get data now that makes sense.
Well, that's good.
Yeah.
That's awesome, though, because I was really tempted to apply to that. So I'm just glad you can say more about your experience and what they have to do, or
and
interactions.
I was talking to them, and they said, "Oh yes, if I can get my tensegrity to work, that maybe they could incorporate it into CompuCell." Would be willing to do something like that. I don't know if it'll work, but at least they figure it's a good platform for integrating other ideas. So that was nice.
Well, great. Yeah, it'd be nice to have like a good platform for tensegrity, I guess.
Well, yeah, at least have it out there, because I think it's a proper way to go about looking at cells, because they're intention.
Yeah. Well, that's good. Thank you. Yeah. Any other updates or? Morgan, did you want to give an update or?
I'm not sure I got anything particularly relevant. No, I mean, check channels for any interesting talks or papers that I might have seen, but that's about it right now.
Yeah, there's a lot of interesting stuff coming up in the Orthogonal Lab Slack that you've been putting in there about different tools and talks and things like that. So, but
I'm not sure that I'm not sure how well grounded they are.
No,
I mean, like super interesting stuff in math and in the formalization in mathematics. Like great sets of talks coming out of that Hausdorff Institute, and they're still coming. They're still dropping videos, even like this morning. I just saw another Kevin Buzzard one too.
Oh, yeah.
But haven't been able to figure out how to connect those back to anything that kind of project related.
Right. Yeah.
Oh, cool.
There was a biological physics webinar by Dapping B on it used CompuCell 3D, and they're looking at the fluidization of tissue, and it was very good. I need to watch it 5 times.
That's great.
I wrote down the guy's name and biological physics, but then I didn't write down the name of the webinar. Anyway, if you want me to, I can find that for you.
Yeah, that'd be great.
Okay, I'll find it and send it.
All right.
You can have a look at it.
Yeah.
Maybe some topic for next week or something.
Yeah, that'd be good.
Okay.
Looks like, yeah, thanks. Looks like Dick posted this origin of life via archaea shaped droplets to archaea. So is this something that was just published? Being printed this week. So that's good. Yeah, that's coming out this week. That's the shaped droplets paper that we featured a long time ago in one of the meetings, and it's basically looking at different shapes of archaea cells and looking at droplets and seeing how they're kind of connected. And so that's good stuff. We'll have to go over that paper as well at some point. So. All right, so I think that's it for today. If we had any other comments before we go. Okay. All right, thanks for attending. Talk to everyone later.
Bye.
Bye.
Take care.
Take care.
Have a good week.
Bye. You too. Bye. All right, now I wanted to cover an article from Asimov Press on breaking the speed limit of cell division or cell cycle. So let's start with the breaking the speed limit on cell division paper. This is from Asimov Press. And this is by Kamal Nahas. So Asimov Press is a really interesting sort of blogging platform, short biological story platform. And so they talk about a lot of issues that are relevant to what we do here. So this is an essay from issue 3. And so this is about breaking the speed limit on cell division. So cell division has a speed. We've talked about this before, and we'll get into this with the papers. There's there are different cycles. There's a cycle of cell division. There are different phases that a cell can be in. And basically it has to be regulated in time. So the cell has to complete one phase to move on to the next phase. And if you extend the amount of time that's in one phase, it can disrupt cell division, and it can disrupt the survivability of a cell line, or it can affect things like differentiation and other properties of the cell. If you increase the rate of cell division, you have faster proliferation of cells. If you slow down cell division, you can basically slow down the proliferation rate of cells. So there are all sorts of things that cell division is relevant to. So the first part of this is computer programmers have it easy. So they talk about computer programmers being able to write a piece of code for just a few minutes before running it and seeing the results. Now contrast this with bioengineers and biologists who do different manipulations of cells and of genes. So biologists manipulate code of a different kind, spending days modifying gene sequences. However, they plod along as they clone plasmids, and so this is something you do in a wet lab environment. We're constrained in biology, as opposed to computer science, by the pace at which cells grow and divide. So in this case, what they're going to talk about is how to increase the speed of cell division to observe things more directly. So due to the nearly ubiquitous use of cloning in life science research, this lost time adds up. And so this is researchers use DNA cloning to make numerous copies of a gene, which can then be inserted into living cells to study how that gene functions or otherwise coaxes the cell to make proteins. So this is important in producing medicines and other types of biomaterials that you want to create using DNA cloning. The ability to clone DNA faster would enable scientists to run experiments, make discoveries, and produce drugs more quickly. So if we can, say, divide, we can help cells divide, and we can create clones of the DNA in a larger and larger cell line, we can actually speed up the amount of time it takes to make medicines or to create a cell line for other various purposes. So that's why we're interested in speeding up cell division, because it speeds up the time that we can get these things that we want to engineer. And so they talk a lot about cloning here. The cloning process itself contributed to a delay in doing the things that we want to do, requiring multiple steps such as cutting and pasting sequences from one plasmid into another, excising antibiotic resistance genes, or flipping sequences around. So when you make genetic manipulations, you often make a manipulation in a single cell, and then you allow that cell to divide. And this is in bacteria. You can then get the copies of that, and you can do things with that line, but you can't make additional edits until you have clones ready to go. And so you have to wait for those clones to sort of generate naturally. For each perturbation, researchers like me must reclone the modified DNA sequences into bacteria, such as E. coli, to mass produce the plasmid. However, E. coli grows slowly, doubling only every 20 minutes under the best conditions, forcing us to wait overnight for bacterial colonies to appear on culture plates. So they're talking about E. coli. They're talking about the doubling time of E. coli, which means that when E. coli is a single cell, it divides into two cells, and then those two cells divide into two cells. So you have 4 cells, and you have this geometric increase, which is, of course, doubling at every generation or every division event. And so when you have, as your colony grows, you get more and more cells, but that's constrained by a doubling time. So the doubling time is how long does it take you to get from a single E. coli cell to 2 E. coli cells to 4 E. coli cells, and so on and so forth. And so that doubling time, as you can imagine, if it takes 20 minutes to go from 1 to 2 cells, then you just keep adding 20 minutes until you get a colony of competent cells, and that could be, a long time if you're looking for, thousands of cells. And so this is something that maybe we have to wait overnight to do instead of having this done in minutes. So if you think of cell division as sort of a compiler for your genetic engineering experiments, it's like running a compiler overnight. And of course, in computer science, that would be unacceptable. But this is something we have to live with in biological engineering. We must then scale up colonies into large populations by culturing them in liquid growth media, adding another 24 hours, after which we can finally isolate the DNA, that's the stuff that you've manipulated, and analyze it. So this would be analogous to a computer programmer taking code, having to put it into the computer and running the job overnight, and then opening up the file and then trying to manipulate or look through the code to see what happened. And so this is something that it's again, it takes a lot of time. The workflow is very it affects the length of the workflow. So they just talk about this in terms of this analogy with computer science. This is a dividing E. coli cell. This is a scanning electron micrograph, and you can see that the cell is dividing here. And of course, this took a certain amount of time to divide. So, it's going to take a lot more time to keep dividing into a competent line with many, many cells where you can harvest them. And of course, you need to harvest them in mass because, a lot of cells, you can't extract DNA out of just like one cell usually. It takes a fair number of cells to get an amount of DNA that you can recognize and analyze. So there are biophysical limits as well. So in recent years, researchers have found biophysical limits on cell division speeds, such as genome size or the time-consuming synthesis of bulky ribosomes. And so this is one of the things about cell division is it not only relies on these different phases, but it relies on the replication of DNA. And so if you have a larger genome, it takes longer to copy all the DNA to create another genome and create another nucleus for that new cell. We also have genes being translated or RNA being translated into amino acids and then made into proteins. And that's constrained by the ribosome and the capacity of the ribosome. And so ribosomes have a certain throughput after which you can't really speed those up. So there's a rate limiting process of the ribosome. And so it takes a long time. If you have a lot of DNA you need to synthesize into a protein, then you're going to have to wait. So there are these mechanisms within the cell itself that slow down the speed of cell division and limit the speed of cell division. And so, making cells divide faster, such as simplifying genomes or ribosomes or enlisting artificial selection to favor genetic mutations that boost speed, are maybe pathways forward, but it's not clear that that's something that you can really speed up that much. So, we might be able to speed up cell division a little bit, but not really to, at orders of magnitude. Comparing the growth speed of different organisms might steer us towards ways of jump-starting this process. The rate at which organisms double is central to how evolutionary biologists weigh up the competitiveness of individuals within a species. Evolutionary fitness boils down to two factors: survival, and of greater relevance here, reproduction. So this is again, this is looking at the fitness of cells, and looking at the survival of cells, some cells over others, and their reproductive capacity. So in bacterial cells, their division rate is basically their reproductive rate. So, some cells will survive over others if there's a certain mutation in a cell, after it divides, then it might make that cell more competent than its sister cell, and it comes to dominate the colony. So, this is a kind of model we can think of in terms of, the kind of colonies that we're producing, and to see, if we can use a mutation-based approach to improving or speeding up cell division, or improving the efficiency of cell division. And so if we compare all life forms by their speed of cell division, multicellular eukaryotic cells reproduce the slowest. So we were just talking about prokaryotes and E. coli. Now, if we consider eukaryotic cells, which are maybe more interesting, especially for biomedical applications, this is something that, it gives us even harder time in terms of speeding up the division rate or the reproduction rate. So multicellular eukaryotic cells reproduce the slowest, and this is because of a number of things, including that you have a lot of organelles that you need to reproduce to have two cells. You have a lot more DNA that needs to be replicated, and so you end up with this situation where you have a much slower rate of division. So-called primary cells harvested from living tissues and cultured in the laboratory can take weeks to grow and spread across the culture flasks. So the doubling time is much slower in these primary cells than eukaryotic organisms. It takes weeks to grow a culture, hampered partly by their large genomes, which take time to copy. It takes about 8 hours for the fastest dividing human cells to double their DNA. So that's even slower. And so speeding up that rate, of course, is going to be a harder lift, because you have a lot of, there's going to be a lot more DNA to replicate, but also you have a lot more structure in the eukaryotic cell to reproduce. In addition, they carry out two intermediate steps between rounds of replication and division, known as G1 phase and G2 phase. So in prokaryotic cells, they don't have these G1 and G2 phases. And so in eukaryotic cells, we have four phases of cell division. So having 4 phases of cell division stalls division by roughly 15 more hours. Primary cells also begin to divide more slowly or come to a halt after a few rounds of doubling, making them inconvenient for long-term experiments. So there's this thing called the Hayflick limit, which is that the number, in some cell lines, you can only divide so many times before you get some sort of apoptosis, which is cell death, programmed cell death, or the lines just don't want to divide anymore. So you have these limits on eukaryotic cells which don't exist in E. coli, and this makes it even harder. It actually slows down cell division rather than speeding up cell division. So in a lot of experiments, people will use rapidly proliferating cancer cells, such as HeLa cells. Actually, HeLa cells are not cancer cells. They're these fused cells. They're like cancer cells and liver cells, and they have different properties all kind of mixed together in the same cell. And so HeLa cells are known as immortal cells, so they can divide indefinitely. They double every 17 hours, so they're very fast growing, and they're very useful for experiments where you need a quick doubling time. But they're not representative of a lot of cells in the body, and indeed, they don't really help you in a lot of contexts where you need to have specific types of cells that you want to work with. Some bacteria, like Mycobacterium tuberculosis, reproduce as slowly as eukaryotes, taking 24 hours to double under the best conditions. However, most bacteria are built for speed. Among the fastest, E. coli and Bacillus subtilis take a mere 20 minutes to divide. So these are pretty fast dividing cells. We talked about E. coli previously. Bacillus is, it's also in this class of very fast dividing cells. Vibrio divides faster, still doubling in only 10 minutes. Bacteria benefit from being single-celled, having tiny genomes hundreds of times smaller than those of eukaryotes, and lacking an intermission between rounds of replication and division. So you have basically in bacterial cells, you have replication division. In eukaryotic cells, you have these G phases that are intermediate to that. And so when looking to design swift cells that can elevate productivity, these microbes make great contenders. What's the delay then? This is where they're considering how to speed this up. So a bacterium's goal is to become bacteria. This is a quote from the late microbiologist Stanley Falkow. So this is the goal of bacteria is to become bacteria. So bacteria will divide. They need to remain dividing to become a bacterial colony and to behave like a bacteria. So this is a movie where they have a time lapse of bacterial growth. It's very rapid, and you get a sense of what they do. They're basically machines that divide and they replicate. That's what they do. That's all they do. It's kind of like C. elegans. They basically eat bacteria and they lay eggs. That's largely what they do. They move around a little bit. And some organisms just do that. They're just replicators, more or less. I don't know if bacteria are just replicators, but that's mostly what they do. So E. coli kind of exemplifies this. They copy their genome every 40 minutes, yet divide in half that time. So they actually divide faster and they copy their genome. So this is interesting because you'd think you'd have to have a full copy of the genome to divide, but in this case, division is such an imperative that it's selected for over copying the genome. And I guess what happens is they just kind of fill in the gaps after they divide. So this is interesting. But this is again this aspect of natural selection at work. In some lines, you can really get rapid division. On other lines, it's very slow, and there are reasons for that have to do with the survival of the cell. They achieve this feat by starting new rounds of replication even before previous ones have finished, juggling up to a dozen rounds at any given time. Unlike eukaryotic cells that pause replication for the majority of the cell cycle, these multitasking bacteria never take a break. By marveling at fast-lane cruisers like E. coli, we witness how evolution favors quickly dividing cells. So this is something that is selected for in evolution in bacteria, this rate of quick division. And so, but yet, despite refinements over 3.5 billion years, hindrances creep in and limit natural selection. In the same way that a race car's tires wear away at faster speeds, cells struggle to indefinitely increase their velocity without having it taking a toll on their health. So this is sort of like the example from HeLa cells. Of course, HeLa cells are these sort of cancer cells that also have properties of liver cells, and they're these immortal cells. But of course, that has an impact on what they do, what they function. And so in bacterial cells, they're very fast dividers, but it takes a toll on what, their sort of their competency and their health, I guess. So this is something that, we always struggle with in natural selection. We have these trade-offs in terms of function that oftentimes limit or constrain, how efficient something can be, or how fast something can be, or even yet how fit something can be. They also can't come up with new strategies from scratch in a way an engineer might. They must retrofit existing genes to adapt, making it difficult to circumvent bottlenecks. So this is a good point that, natural selection doesn't operate like, engineer might optimize something. You have to work with the existing genes that you've given, and you may be able to retrofit existing genes to adapt. But sometimes you have these bottlenecks that, are really kind of hard to overcome because you don't have the raw material of evolution, to overcome them. So, sometimes, we talk about mutations being sort of like a way to get adaptations, but sometimes, those adaptations kind of, there has to be something there. And so when the cells are dividing like that, they may have enough mutations to overcome these bottlenecks. They may not. Genome size creates the first bottleneck to cell division. So the genome size, of course, is important to kind of speeding up cell division. So if you want to have fast cell division, you generally want to have a small genome. But, of course, you can divide faster than you can replicate DNA. So there's that. But, of course, at some point, the genome size becomes a constraint. And again, large genomes generally take longer to copy because as a DNA polymerase enzyme runs across DNA strands during replication, it pauses to proofread the copied sequences. So this whole process of copying DNA. You have a polymerase runs along the DNA strand. It's proofing the copy because it can introduce harmful mutations, and so it needs to do those kinds of checks and then assemble into a new double helix. Evolution hits the brakes to avoid burning its tires. If polymerases rush this phase, the cell might accrue many harmful mutations. So you can accrue random mutations during cell division, during replication, but you can also accrue harmful mutations. Most mutations that sort of, get passed on are neutral. You're going to get harmful mutations. Those usually get edited out. And of course, it's imperative for a competent cell line to have these harmful mutations edited out at the source. So that's what happens with cell division. And that's what slows down cell division in part. Although the rates vary, a prokaryotic polymerase copies 1,000 bases per second on average, but a eukaryotic one copies only 30 in the same time. And so this is where you have this sort of this speed of the polymerase. In prokaryotes, it's much faster, and so we have this speed, differential. Those slower eukaryotic polymerases make up for this by initiating replication at multiple sites along chromosomes, thus dividing the job between multiple enzymes. Bacteria doesn't split up this task in the same way. They begin replication at just one locus, with two polymerases running in opposite directions to copy the whole genome. So this is where, we have this, eukaryotes have developed or evolved these different mechanisms for speeding up cell division because, eukaryotic cell division could be infinitely slow otherwise. But still, it's slower than prokaryotic cell division. And so this talks about proofreading and kind of what the mechanisms are there. The rate at which polymerase enzymes fulfill their function illustrates that genome size acts as a fundamental barrier to speedy replication. Smaller genomes, therefore, could lead to faster growing cells. So these are the ribosome subunits, which is the other bottleneck. And so again, RNAs that get transcribed from genomes, they go through a ribosome, they get translated into amino acids, which get assembled into proteins, which are needed for a lot of the structural aspects of the cell. And so, but ribosomes have a certain rate that they can translate things. So they have this sort of, it's like feeding things into a paper shredder. or feeding things into a tape machine. You can't overload it with stuff. So there's this whole literature on the speed of ribosomes, and that's where your other constraint comes in. So the biosynthesis of ribosomes, the complexes that build all the cell's proteins, cause an additional bottleneck. Ribosomes create all the enzymes needed to catalyze chemical reactions important for growth, and ramping up enzyme production could, in theory, speed up cell division times. However, this would require the cell to make many more ribosomes, which in turn would slow down division speeds, because the production of these behemoth complexes, in the cellular sense, they're not very big, requires copious resources and time. So, relative to sort of DNA replication, these are huge, but they're not that big in the cell. But making more of them per cell slows down division speeds. And of course, there are more complex ribosomes in eukaryotic cells than in prokaryotic cells, which of course slows down their division rate. For a 2021 paper, researchers at the California Institute of Technology estimated how long it would take for E. coli to churn out a single ribosome using a back-of-the-envelope calculation. So they just wanted to know, from first principles, how long does it take to churn out a single ribosome in E. coli. RNA and proteins comprise each ribosome. The cell synthesizes these two biomolecules at different rates. The RNA polymerase enzyme transcribes DNA into RNA at a rate of approximately 40 bases per second, but proteins emerge much more slowly. Only around 15 amino acids are added to a growing protein chain in the same amount of time. With approximately 50 proteins in a ribosome, totaling approximately 7,500 amino acids, they estimated that ribosomal protein synthesis is one of the slowest processes within E. coli. So it takes 8 minutes for a cell to synthesize the proteins needed to make just one ribosome. A significant constraint. So this is our minimal cell that we've talked about in meetings in the past. This is a JCVI minimal cell where you have the molecular modeling and you have the sort of the coarse-grained cell modeling, and you're putting all that together. And so this is where they can kind of do these kind of experiments. They can simulate these things. We might want to ask the question, if E. coli possessed only one ribosome, how long would it take the cell to divide? If this one ribosome added 15 amino acids to growing protein chains every second, and if total protein content of the cell amounted to 1 billion linked amino acids, it would take over 2 years to build up all the cell's proteins. So, this is kind of interesting because, this really tells you something about like this kind of speed of cell division and what's all involved in building a cell. So, we can do these back of the envelope calculations. We can do these simulations, these molecular dynamic simulations, these whole cell simulations. But this is only part of the equation. So when E. coli divides every 20 minutes, then, because it spreads its molecular labor across anywhere from 10,000 to 100,000 ribosomes. These estimates match experimentally determined ribosome concentrations, suggesting that ribosomes already work at maximum capacity in these microbes. When ribosomal proteins evolved, they became integral to cell survival. But as cellular life sped up, synthesizing these proteins became a time-consuming burden that cells couldn't abandon without losing other crucial functions. So this is interesting because cells can do other things, right? Like an E. coli could do other things. But the imperative is to divide. So as its imperative is to divide, it sort of devotes all of its resources to the mechanisms for dividing. So there's this aspect of needing to sort of speed up, but also needing to have like DNA replicated and things pass through ribosomes to produce proteins. Ribosome synthesis may take most credit for limiting division rates, but the cell has yet other bottlenecks to contend with. So we've talked about ribosome synthesis and genome length, which are two constraints on division rate. So this slows down division rate. But we have other bottlenecks that we have to consider. So in 2011, biophysicists at Stony Brook University estimated how the rates of protein folding impacts E. coli growth. They found that some proteins fold much slower than others, depending on their three-dimensional structures. Helical twists in a protein form faster than parallel running strands called beta sheets, for example. Some proteins take as much time to fold as an entire E. coli takes to divide. This suggests that even if cells could sidetrack the time-consuming synthesis of ribosomes, slow protein folding could be another roadblock to faster division. So protein folding rates are important. Of course, proteins need to fold in a certain way at a certain rate in order to become fully functional. So protein folding usually doesn't take a long time, but sometimes they can fold slowly. And if they have a more complex structure, then that's going to take longer to get those in place. So finally, we can ask the question, how do we make cells go faster? So decluttering the genome is one answer. We can just remove all the extraneous genes and leave behind only the bare essentials. So we have these what we call minimalist microbes, and these are these minimal genomes. So there's the GCVI SYN3A, which is the one that we just saw with the minimal cell here. This is based on this kind of minimal genome. And, that minimal cell is half of the original size of the genome. So we have a bacterium that has, we have like a certain number of genes in the wild type. We reduce that by half, and we get this minimal cell. It's minimally competent as well. So they don't always survive, but they are living. They have all the essential cell genes they need to, reproduce, which is their imperative, but they have a minimal set of genes. So this is not like, this only gives you the ability to live. The blueprint of a minimal cell, mini bacillus, and this is, of course, this paper where they do this in bacillus, where we have analyzed the genome of B. subtilis and selected all genes that are required to live life in complex medium at 37 degrees Celsius. The selection is based on known information on essential genes and functions, as well as on gene and protein expression data and gene conservation. The list presented here includes 523 and 119 genes coding for proteins and RNAs, respectively. These proteins and RNAs are required for the basic functions of life and information processing, so that's replication and chromosome maintenance, transcription, translation, protein folding, and secretion. So these are the key behaviors. You don't really get anything else, but you need these basic biochemical mechanisms to survive. You also need metabolism, cell division, the integrity of the minimal cell, which means just having a structural home for the other parts of the cell. The completeness of the selected metabolic pathways, reactions, and enzymes was verified by the development of a model of metabolism in the minimal cell. A comparison of mini bacillus genome to the recently reported designed minimal genome of Mycoplasma mycoides, which is the GCVI SYN 3.0, indicates excellent agreement in the information processing pathways, whereas each species has a metabolism that reflects specific evolution and adaptation. The blueprint of mini bacillus presented here serves as a starting point for a successful reduction of the B. subtilis genome. So this is where they want to reduce this genome so much that they can really kind of get it down to the core genes. It survives but does little else. It just produces enough to have a cell that isn't, dead on arrival, or can actually reproduce itself. So this is, really kind of getting down to the bare minimum. And so even then, we still have problems with getting cell division to go faster. We can get these minimal genomes, but it still takes time not only to replicate the genome, but build all of the proteins that are needed to build a whole cell. These case studies warn that minimizing genomes don't necessarily improve growth rates, even though it should be theoretically possible. And so in E. coli, we have one origin of replication in wild type, but synthetic biologists at Yonsei University in South Korea found that by adding 2 extra origins at different sites in the chromosome could propel cell division. So what they're talking about here is that E. coli, of course, is a circular genome, and there's one site, origin site or origin fork for replication, and so it starts at that point and goes around, and it takes a while to go around and with the polymerase and make a copy. But if you have 2 extra origin sites, at different sites on the chromosome, you can actually copy the genome in less time and speed up cell division. So they worked with a low dividing E. coli strain called MG1655, which normally takes 1 hour to double up. By adding 2 more origins, they shaved 4 minutes off the doubling time. So this is probably, trivial in terms of doubling time, because you would think it might be, cut it by a third, but it only shaves 4 minutes off the doubling time. Though they only achieved a small boost, the data is encouraging. The cells were viable despite carrying extra origins, and the findings revealed that tweaking the number and position of these replication start sites would speed up cell division rates. It'd be interesting to see if adding surplus origins in fast-dividing E. coli strains could achieve a similar effect. We could also move genes around. So scientists recently found that replication influences the order in which genes of the bacterial chromosome are expressed. So during a replication cycle in E. coli, for example, genes are switched on in the order they are copied, starting with genes located near the origin, thus producing extra copies of these genes sooner and boosting their expression. So you can actually create maybe more copies of a gene to boost expression of genes, of that gene, or you could shuffle genes around so that they would position those that accelerate growth near the origin. So again, you can select for certain types of genes through replication and play around with that. So this is interesting. And so for a 2021 study, researchers used minimal mycoplasma cells to pinpoint genes essential for cell division, finding 7. So there's 7 essential cells, or the 7 essential genes in sort of promoting cell division. The most notable genes include FITZ and SEPF, and FITZ, of course, is involved in the ring that sort of pushes the cell into 2 different cells. So it does this budding where it produces this protein ring, and then it contracts, and it shifts the single cell into 2 cells. It allows the cell to sort of divide into 2 parts. FITZ and SEPF encode proteins that split a dividing cell in 2 by assembling into a constricting ring that tightens and splits apart the cell membrane at the dividing line between budding cells. Without this cell splitting machinery, mycoplasma cells inflate and swell, sometimes up to 25 times their typical volume, but do not divide. So this is important that we have these genes in place. So this is an example here of FITZ proteins forming this contractile ring, and just kind of squeezes the cell body into 2 parts. And then, of course, it divides into 2 cells. The DNA migrates into each copy, and that's what we have. So this is a molecular model showing FITZ proteins in green and red here, extracted from a microbe constricting a liposome, which is blue. So the liposome is the membrane of the cell, and it's constricting it into 2 different compartments. Additionally, rather than trimming genomes, perhaps doubling times could be reduced by trimming down ribosomes. So maybe we could have fewer ribosomes. But remember, we need more ribosomes to have more proteins produced. So that's always going to be a trade-off. Recall that a typical ribosome in bacteria is built for 50 proteins and 3 strands of RNA. It is the RNA that catalyzes chemical reactions. Structural biologists have already designed blueprints for a minimalist yet functional ribosome. So we're not only looking at minimal cells, but we're looking at these minimal ribosomes. Linear ribosomes might take less time to synthesize and therefore boost speeds. So this is where, we're getting into a lot of these minimal models and seeing what kind of is the lower bound of the speed. How fast can we divide? And so the researchers in this study up here, which I won't get into, identified 27 ribosomal proteins not conserved across bacteria, archaea, and eukaryotes. How much time could the cell save in synthesizing ribosomes if it relinquished these components that are not conserved? So this is where, we're kind of getting rid of all the adaptations and we're getting like a core ribosome structure. To match these earlier estimates made by researchers at Caltech, if you assume the ribosome is made up of 50 proteins of equal size, totaling 7,500 amino acids, then these 27 would account for roughly 4,050 amino acids. At a rate of 15 amino acids added to the growing protein per second, researchers could theoretically shave off about 4 minutes, half the time taken to synthesize a ribosome. So now we have this 4 minutes time savings again, but instead of in the genome, it's in the ribosome. And so this is actually about half the time it takes to synthesize a ribosome. So this is, again, we can shave time off of this process. In this case, it's a little bit more, relatively speaking, it's a little bit more significant. But this is, of course, there are problems with this in practice. Some of these proteins may prove crucial to E. coli ribosomes to function, even if they're not conserved across the 3 domains of life. So reducing the ribosome to this scale might not work. And again, you're just trying to find sort of the common ancestral ribosome. But of course, in the context of the cell, it might be that E. coli needs these specialized ribosomes. And that's another thing that we have to consider with minimal cells. What can we reduce without killing the line or making it non-competent? And so finally, there's this section going off-road. So this is more of this sort of back of the envelope calculations of different systems, looking at this organism called Vibrio natrigens. This microbe's hasty division has recently drawn the attention of scientists. With a minimal doubling time of 9.4 minutes, twice the speed of E. coli, it holds the place as one of the fastest dividing organisms known to science. Colonies of V nitrogens appear on agar plates in as little as 6 hours, as opposed to 14 hours for E. coli. Unlike Clostridium perfringens divides faster, doubling to approximately every 6 minutes. However, Clostridium perfringens proves tricky to work with in the lab. So this is this Clostridium, which is hard to work with. So we would like to have this Vibrio natrigens as the model organism. For one thing, being strictly anaerobic, it does not survive well when exposed to oxygen. Plus, it's a human pathogen, so it's hard to handle. But V nitrogens, of course, has none of these limitations. It's easy to culture and modify. And so at Synthetic Genomics in California, they've minimized its genome for industrial purposes, excluding 194,000 DNA bases. So they don't delete the genes one at a time. They don't do these combinatorial knockout experiments in this case. What they did here is to compare the chromosomes of 2 V nitrogens strains side by side. This allowed them to spot differences in their genomes, highlighting regions that weren't conserved and could be jettisoned. So we're only interested in the conserved regions between individual cells or cell lines. And so unlike in the JVCI Syn3A mini bacillus models, where they do these combinatorial knockout experiments where they get rid of genes that are non-essential, they actually just get rid of DNA bases that are not conserved. And so now you're actually discarding genes that encode virus sensing machinery. And so these are things that you don't need in the lab. There are all sorts of things that you remove that may be needed in nature, but aren't needed in a very fixed environment. So this makes it easier to engineer, but also it makes it faster to divide. So we're trying to reduce the time it takes to divide. And so this is where, we just have to imagine, how efficient can we make these systems? How fast can we speed up cell division? And when we're working in the lab, we don't really need to have a robust genome. We just need to have something that divides quickly. So if we can engineer that, then we're better off. And so we'll probably never match the speed of computer programming, but we can improve using model organisms and some of these bioengineering techniques. So hopefully you found that interesting. Thanks for paying attention, and I hope you learned something.